Hello everyone, I'm your anatomy mentor, Dr. Hum, and today I will be discussing the histology of the cerebellum. Now, you know the cerebellum is an important part of our brain. We can say it is the smaller version of our cerebral hemispheres. It is located in the posterior part of the skull, and this is a very important organ because the cerebellum it helps us in coordinating the voluntary movements and also maintaining our equilibrium and posture. So this cerebellum, today we shall be discussing its histology, that is, we'll be talking about the layers in the cortex of the cerebellum and the different cell types which are present in these layers. I'll also discuss the connections of the various types of cells within the cerebellar cortex and a little mentioning of the intracerebellar nuclei. So let's start with today's topic. We see where is the cerebellum located. I just told you that this is a smaller version of our cerebral hemispheres. There are two large ovoid-shaped cerebellar hemispheres interconnected by the narrow contracted region in the middle, which is the vermis. Now, these cerebellar hemispheres and vermis, they form this part of the brain, which is the cerebellum. And it is located in the posterior cranial fossa, and it is lying behind the fourth ventricle, pons, and upper part of medulla oblongata. So, like I told you, its function is coordination of voluntary movements, maintenance of posture and balance, and also it is responsible for the maintenance of the tone of the muscles. So before beginning specifically with the cerebellum, generally speaking, you should know that whenever we come to the gray matter is present on the outer aspect, and there it is known as the cortex, whereas the white matter is present in the inner part, and there it is known as the medulla. The cortex contains nerve cell bodies, axons, dendrites, and glial cells, and it also is the site of numerous synapses. Whereas the medulla is the part which only has axons of nerve cells, plus some supporting glial cells and blood vessels. Now let's talk about the cerebellum. Our cerebellum consists of the outer covering of gray matter with the central core of white matter, which also further contains these deeper collections of gray matter known as the nuclei, which are 4 pairs. Now, the cerebellar cortex, which is the outer gray matter, this is present in the form of folds, which are known as folia. The word folium literally means a leaf. And these folia, they are supported by the branching internal white matter. Now, the point to remember is that in comparison to the cerebral cortex, when we talk about cerebellar cortex, here the structure is uniform and it is not different in any region. And this is a particular feature to be kept in mind when we talk about the cerebellar cortex. There are 3 layers in the cerebellar cortex. These are the 3 layers of gray matter. The outermost one, that is known as the molecular layer, this has relatively few neurons and larger number of unmyelinated fibers. Then deeper to that is the Purkinje cell layer, which consists of a single layer of large flask-shaped neurons. And in the deeper aspect is the granule cell layer, which has very small neurons densely packed together. So let's look at the histomicrographs of the cerebellar cortex. On the left in the histomicrograph A, we can see at the lower magnification that these are the folia, which I just mentioned, or the enfoldings of the gray matter of cerebellar cortex. And these are, you can see, these folia, and this shows us the gray matter and the layers of the gray matter. And internal to the gray matter is the branching white matter of the cerebellar region. Now let's look at the histomicrograph B, which is at a higher magnification. Remember, these sections are all stained with hematoxylin and eosine. And over here in this higher magnification, we can see the 3 individual layers of cerebellar cortex. The outermost or most superficial one is the molecular layer, which I told you contains relatively fewer neurons but more nerve fibers. Beneath that, then we have this here, Purkinje cell layer, which is a layer of flask-shaped neurons in a single file. And beneath that, then you can see is the darker staining layer, which is actually the granule cell layer, and it appears to be darker because of the neurons which are densely packed together. And this gives the bluish appearance of the granule cell layer. This is the innermost part of the gray matter. And at a still higher magnification, we can see that this molecular layer, it has a few neurons. These nuclei of these neurons have been stained basophilic, and you can see in between these neurons is the larger quantity of the nerve fibers. Also at this higher magnification, we can see the second layer, which is the Purkinje cell layer. We can see they have the neurons, which are the large ones with the prominent rounded bodies, and we say they're like a flask shape. And inner to that, here comes the granule cell layer, which consists of small to medium-sized neurons, and they're densely packed together. So the nuclei, they are taking on this basophilic, prominently basophilic stain. Now, let's talk about these layers one by one. The most superficial layer of the gray cortex of cerebellum, which is the molecular layer, it contains extensively branched dendrites of the Purkinje cells and the non-myelinated axons of granule cells, which pass upward into this molecular layer. And these axons of granule cells, as they enter molecular layer, they will bifurcate, that is, they will divide into two parts to run parallel to the surface, and they will eventually synapse with the dendrites of the Purkinje cells. Remember, within the molecular layer, there's also another cell population, the stellate and basket cells. Now coming to the inner second layer of the gray matter of cerebellar cortex, which is the Purkinje cell layer. Keep in mind, these cells were the first neurons to be identified, and they were the first neurons to be analyzed in microscopic detail. So that's a little historical relevance of the Purkinje cells. Remember, they are the large Golgi type 2, type 1 neurons, and so their cell bodies are the most prominent, most conspicuous in the cerebellar cortex. They have a clear vesicular nucleus with a deeply staining nucleolus, which is known as the owl's eye appearance in the stained histomicrographs, and also it has irregular Nissl granules. Purkinje cells, they have their dendrites, which are going to be visualized in the molecular layer, and this is the branching, which is known as the arborization, but their axon is not easily visible in the routine H&E stain sections. The fan-like dendrites, they are oriented at right angles to the long axis of the folia. The dendritic spines, which are covering the primary and secondary branches of the dendrites of Purkinje cells, synapse with the granule cell axons in the molecular layer. And what about the efferents? The efferent fibers, that is the fibers which are leaving the cerebellar cortex. Remember, they are the axons of the Purkinje cells that will pass through the granule cell layer, and then they will go deeper into the white matter to synapse with the central nuclei of the cerebellum. A few Purkinje cell axons will also bypass the central nuclei of cerebellum to directly end or synapse in the vestibular nuclei of the brainstem. So the Purkinje cells also, there are some collateral branches which make synaptic contacts with the other cells in the granular layer. And the entire output of the cerebellar cortex conveyed by Purkinje cell axons, keep in mind their output is inhibitory. The principal neurotransmitter, therefore, is GABA-aminobutyric acid. Now let's come to the innermost layer of the gray matter of cerebellar cortex, which is the granule cell layer. Well, like I showed you in the histomicrograph that were stained with hematoxin and eosine, there is a spotted blue appearance of the granule cell layer because of the large quantity of these small nuclei of granule cells, which are densely stained, and they have scanty cytoplasm. Over here, they contain glutamate as the neurotransmitter, and the dendrites of granule cells they make contact with the mossy fiber input, whereas the granule cells receive incoming impulses through the input from other parts of central nervous system. So remember, when we talk about granule cells, we have the axon of the granule cell, which is going to enter the molecular layer superficially and bifurcate into these parallel fibers. And these parallel fibers, remember, they are running at right angles to the planes of the dendritic trees of Purkinje cells. So as a result, each parallel fiber synapses with dendrites of numerous Purkinje cells, and it also synapses with the basket and stellate cells. Now, there is another cell population in granule cell layer, which are the Golgi cells. They are larger in size than the granule cells, and they're also scattered within the superficial part of the granule cell layer. The dendrites of these cells, they will ramify or branch in the molecular layer, and their axons will terminate into branches that synapse with the dendrites of granule cells. Now, here is a pictorial representation of the layers of the cerebellar cortex, that is the gray matter, and we're going to look at the different cell types within the gray matter of the cerebellar cortex. Like I told you, there are three basic layers. The outermost or most superficial one is this here molecular layer, and then beneath that is the layer of the Purkinje cells. These are the most conspicuous or prominent cells in the cerebellar gray matter, which helps us in identifying cerebellar cortex. And they are present in a single file or a single row. Beneath this layer, the deepest part of the gray matter of molecular layer is the granular layer. And over here now, let's look at the different cell types which are present. First, let's keep in mind that these Purkinje cells, their dendrites, they are going to go superficially into the molecular layer and they undergo extensive branching. This is known as arborization. And the axon, it exits from the lower aspect of the Purkinje cell, passing through the deeper layer. It is the one which exits the cerebellum, and this is the one, remember, which is going to have an inhibitory function, and it is going to synapse. Most of these fibers are going to synapse on the deeper cerebellar nuclei. A few fibers will exit without synapsing on the cerebellar nuclei, those which are going to the vestibular nuclei of the brainstem. But majority will synapse on the cerebellar nuclei. And as I told you in our routine hematoxin eosine stain sections, this axon is not so easily visible, but the dendrites and the cell body of the Purkinje cells, they are easily identified. Okay, so then let's come to over here the inner layer. The inner layer, which is the granule cell layer, has granule cells, and these granule cells, you can see their axon. It is going to go superficially in the molecular layer. It is going to bifurcate into these parallel fibers, which will then synapse with the dendrites of the Purkinje cells. Whereas on the inner aspect, the dendrites emerging from the granule cell will form synapse with the mossy fibers which are entering the cerebellar cortex. There's another cell type in the granule cell layer, that is the Golgi cells. These Golgi cells have their dendrites entering, ramifying over here, branching in the molecular layer of cerebellar cortex, whereas the axons, they will synapse on the with the dendrites of granule cells, and the dendrites of granule cells along with the axons of Golgi cells and the inner synaptic terminal of the mossy fibers, they will collectively form a structure which is known as the glomerulus. We will be talking about the glomerulus and looking at its pictures in detail in the further slides. Now let's look at the other cell types in the molecular layer over here in the gray cortex. We've got the stellate cells also and the basket cells. Well, the stellate cells and the basket cells, you can see that both of their dendrites, they will synapse with the axons of the granule cells. Also, the axons of stellate cells will synapse with the dendrites of the Purkinje cell, and the axons of the basket cell will wind around the body of the Purkinje cell in the form of a network, also called a basket. Therefore, that's why we call them the basket cells. When we're talking about the incoming fibers, we have two types of fibers which enter our cerebellar cortex. One are the mossy fibers, and which I told you form the glomerulus, but also we have these climbing fibers which will form synaptic contacts with the dendrites of the Purkinje cell of the molecular layer. So this was a detailed explanation of the different cell types and their connections within the cerebellar cortex. Okay, so like I said, the main input to cerebellar cortex is through the climbing and the mossy fibers. This is an excitatory input. And remember, the climbing fibers are terminal fibers of olivocerebellar tracts, whereas the mossy fibers are the terminal fibers of all the other cerebellar afferent tracts, namely vestibulocerebellar, pontocerebellar, spinocerebellar. Climbing fibers, they make synaptic contact with the dendrites of Purkinje cells. And remember that one Purkinje neuron makes synaptic contact with only one climbing fiber, but one climbing fiber makes contact with several, up to 10 Purkinje neurons. Now, regarding the mossy fibers, they synapse with granule cells, and through the granule cells, indirectly, a single mossy fiber may stimulate thousands of Purkinje cells. So they have a more diffuse excitatory effect than the climbing fibers. Now, here is a histomicrograph, which is the neutral red stain on a high power. And like I told you, we can see the cell bodies of these Purkinje cells. And we can also see the dendrites, which are emerging from Purkinje cells and entering this superficial molecular layer of gray cortex of the cerebellum. Although the axon of the Purkinje cells, they are not visible in this histomicrograph. Let's look at the deepest layer of the gray cortex. That is the granule cell layer, which has these densely packed small sized neurons with the scanty cytoplasm. These are the granule cells. Whereas the most superficial one, the molecular layer, it has a few neurons, but mostly it contains the unmyelinated nerve fibers. The cell population in the molecular layer, which are the stellate cells, they have their characteristic feature that their dendrite synapse with parallel fibers of granule cells, while their axon synapse with the dendrites of Purkinje cells. So these are small star-shaped cells in the molecular layer. The basket cells, they are lying in the deeper part of molecular layer of cerebellar cortex. And as I mentioned before, their dendrites synapse with parallel fibers of granule cells, while their axons form networks in the shape of baskets around the cell bodies of the Purkinje cells. Now, the Golgi cells, which was a cell population lying in granule cell layer, their dendrites enter molecular layer to synapse with the parallel fibers of granule cells. And as I mentioned before, their axons, they branch and they permeate granule cell layer to take part in the formation of a structure known as a glomerulus, the plural of which are glomeruli. So what is the function of these additional neurons in the gray matter of cerebellar cortex, namely the stellate, basket, and Golgi cells? Well, they are inhibitory interneurons, which means that they limit the area of cortex excited and they influence the degree of Purkinje cell excitation by the climbing and mossy fibers. Now here we have this organization. This is another pictorial representation showing the connections of different cell types that are present within the cerebellar gray cortex. Over here, these are the three layers of the cerebellar cortex. From the superficial to inner aspect, we can see most superficial is molecular layer. Then comes our Purkinje cell layer, and then the deeper one, which is the granular layer. And beneath that is the white matter of the cerebellum. So here we can see these Purkinje cells, the flower-shaped neurons present in a single file. the dendrites are emerging from their superficial aspect and ramifying within the molecular layer, whereas the axon, it passes through the deeper layers to leave the cerebellar cortex. So the dendrites of Purkinje cells, they will synapse with these here parallel fibers, which are the bifurcations of the axons of granule cells, which are present within the granule cell layer. The axons which are emerging from the basal aspect of the Purkinje cells, it forms the efferent fibers of the cerebellum, and it also, remember, has the inhibitory function. So over here, let's look at these granule cells. Let's look at the dendrites of granule cells. They are synapsing with the mossy fiber and also with the axon of the Golgi cells, which are present in granule cell layer. And this collectively forms a structure which is known as the glomerulus. So the climbing and mossy fibers, they are the afferents which are entering within the cerebellar cortex. The mossy fiber participates in formation of glomerulus in granule cell layer, whereas the climbing fiber, as the name indicates, it is going to pass through these layers of the cerebellar cortex to synapse with the dendrites of the Purkinje cell. So let's talk a little about the glomeruli. There are spaces in the granular layer. If we look at the histomicrograph of a granular layer, we can see that there are certain areas where granule cells are not present. These spaces are known as cerebellar islands, and they are occupied by the specialized synaptic structures known as glomeruli. So here is a detailed structure of a glomerulus. They are complex synaptic structures. The core is formed by the expanded terminal end of the mossy fiber, which is known as the rosette. And upon this rosette, numerous dendrites of granule cells are going to synapse, and this glomerulus also receives the axon of the Golgi cell. Now, remember that these synapses are inhibitory, and this entire glomerulus, which is about 10 microns in diameter, is surrounded by a neuroglial capsule. So in the histomicrograph of the cerebellar cortex, here we can easily identify the Purkinje cells. Superficial to that, over here, this is the superficial layer, which is the molecular layer. We can see a few neuronal bodies, and we can see the basket cells, the neuronal bodies, and also we can see this here, most of the areas occupied by the unmyelinated nerve fibers. Also within the molecular layer, we can see the dendrites of the Purkinje cells. Now, in the deeper aspect of the gray matter of the cerebellar cortex, this here is the spotty blue appearance that I mentioned of the granule cell layer because of the densely packed small-sized neurons, which are the granule cells. But there are certain areas where the arrows are pointing towards the areas where we do not see the granule cells. These empty areas are the lightly stained areas which contain the glomeruli, the structures which I just explained to you, which were the specialized synaptic structures. Okay, so keep in mind that the stellate and basket cells are the ones present in molecular layer. Purkinje cells are obviously in the Purkinje cell layer, and these are the two cell types, granule and Golgi cells, which are present in the granular layer. And within the white matter of the cerebellar cortex, I told you, are the intracerebellar nuclei, which are masses of gray matter, and from the lateral to medial aspect, these are 4 pairs of nuclei. The first one is the dentate nuclei. This is gray matter folded upon itself in the form of a crumpled purse with an open end facing medially. And then are the emboliform and globose nuclei. These two can be collectively called the nucleus interpositus. And then the most medial one, which is close to the midline, is the vestigial nucleus. So these are the intracerebellar nuclei, and they are lying close to the roof of the fourth ventricle, so they're also called the roof nuclei. Now I'm going to discuss, based on the physiological or functional aspect, the neuronal circuitry of the cerebellum. So let's look over here at the input, the afferent fibers of the cerebellum. These are the climbing fibers and the mossy fibers. The climbing fibers, they are going to pass through the layers of cerebellar cortex and synapse on the dendrites of Purkinje cells. And their input, as the plus sign shows us, is excitatory. They also give some collaterals or sideway branches to the deeper nuclei. Now, the other type of incoming fibers, the afferents, are the mossy afferents. And these mossy afferents, you can see they are going to form this structure. They're going to form a synapse on this synaptic structure known as glomerulus, which is present in the granule cell layer. And over here, it synapses with the granule cells. And you can see then again, plus indicates that the input is excitatory. And these granule cells, their fibers, they have the processes which are the axons, which are going to then enter the molecular layer and form their connections with the dendrites of Purkinje cells, which is still the excitatory sort of connection. Okay, then now let's come to this Purkinje cell. These Purkinje cells, their axons are emerging from their basal aspect, and then it is going to synapse on the deeper nuclei. As I told you, these are the efferents emerging from the axons of the Purkinje cells are the efferents, and their signals are, as can be seen by this negative symbol, an inhibitory effect. So their effect is inhibitory, and they synapse on this deep nuclei, which themselves are having the excitatory effect. So this is how the Purkinje cell is going to modulate the actions or the signals which are emerging from the deep nuclei. Whereas a few axons of the Purkinje cell will pass without synapsing on deeper nuclei. They're going directly to the vestibular nuclei in the brainstem and having their inhibitory effect over there on the vestibular nuclei. So vestibular nuclei, according to some neurologists, might be considered, according to some histologists, as the displaced nuclei of the cerebellum. Okay, so the efferent fibers, let's summarize. They're axons of Purkinje cells, and they are the those axons which terminate in cerebellar nuclei, but some efferents from flocculonodular lobe bypass cerebellar nuclei and terminate directly in vestibular nuclei in the brainstem. And the fibers from dentate, emboliform, and globose nuclei leave the cerebellum through the superior cerebellar peduncle. But the fibers from the fastigial nucleus, they leave the cerebellum through the inferior cerebellar peduncle. So that concludes with the basic histology of the cerebellar cortex, and just a little bit of a clinical correlation. There is the condition which is known as cerebellar ataxia. If there is a disease that damages cerebellar cortex, and this damage can occur due to a trauma or tumor or some neurodegenerative diseases, and also it can be drug related due to alcohol or hypoxic damage. But in any case, when the cerebellar cortex is damaged, the condition known as cerebellar ataxia can occur. Ataxia, as we know, the normal function of cerebellum is to maintain posture, balance, and equilibrium. So when this function is disturbed, there will be ataxia, and ataxia is the sign in which there is poor coordination of voluntary movement and there is loss of balance. So that is one of the clinical outcomes of cerebellar disease. So if you found today's discussion on the cerebellar histology helpful, then don't forget to like and subscribe to my YouTube channel, AnatomyMentor. You can do that by just clicking on my picture, which is going to appear at the end of this video. And also don't forget to hit the notification icon, the bell icon, which is going to also appear below this video. So thank you for listening to today's topic, and for further topics related also to neuroanatomy, embryology, and gross anatomy, keep waiting for my lectures, and don't forget to hit the notification button. So take care, enjoy your studies, and see you next time as I come with another video presentation. Take care.